in an experiment.
We don't know yet.
Why is light so fast?
Like, it sounds so simple.
They had no idea.
But now the data speaks.
I find this not only refreshing, but at some level astounding.
Nature.
Welcome back to the Nature Podcast. This week, a gamma-ray producing microquasar.
And how a smart insulin molecule could help people with diabetes. I'm Nick Petrichow.
And I'm Benjamin Thompson. This week, physicists have identified a strange gamma-ray producing object in the Milky Way, which could help researchers in their search for the source of galactic cosmic rays, something which has left them scratching their heads for years. And this source is something quite unexpected, a microquasar.
So you probably could tell from the name. The microquasar is a micro size of quasar. They have black holes and they have a companion star.
This is Xiaojie Wang, one of the authors of the new work. It's a bit of a journey from microquasar to galactic cosmic rays, but stick with me, we'll get there. Now, regular quasars are a rare type of supermassive black hole that fires out enormous jets of matter. But microquasars are kind of their much smaller sibling. Microquasars are binary systems. They're made up of a very dense object, maybe a neutron star, but most likely a black hole, that strips material from an orbiting star before firing it out in much smaller jets of subatomic particles. To date, not many of these microversions have been identified in our galactic neighborhood.
In the Milky Way, the known microquasars is about 20-ish.
Of these 20-ish, one stands out as a bit of an oddity, as it's firing out gamma rays. For quite a while, this was thought to be a cosmic anomaly, but now Xiaojie and her colleagues have found evidence of another microquasar that's pumping out gamma rays as well. It's called V4641 Sagittarii. Now we are in an n equals 2 situation, suggesting that gamma-ray producing microquasars could be more common than previously thought. They found this new gamma-ray source by working backwards from a signal detected by a telescope called the High-Altitude Water Cherenkov Gamma-Ray Observatory, or HAWK.
It is different with other telescopes. It is consistent of a bunch of water tanks. And inside of water tanks, we have the photosensors to collect the signals from the deep universe. And our main object will be the gamma rays.
These 300 water tanks at high altitude next to a Mexican volcano look like grain silos, but they're key to working out where gamma rays come from. But the telescope doesn't detect them directly. Rather, it detects particles created by gamma rays as they interact with Earth's atmosphere. These particles rain down, causing tiny flashes of light in the water tanks. These flashes contain information that allows the researchers to work backwards, to figure out where in the atmosphere these particles came from, and so where the gamma rays hit. They can then trace those back through space. And when Xiaojie and her colleagues did this, they found a region of the sky where the gamma rays were likely to have come from, leaving Xiaojie to check other observations of that region taken using telescopes that look at different parts of the spectrum to work out what's there and narrow down the source.
So I check if there any X-ray detection there, if there optical detection there, and how about the radio, how about the infrared, other instruments have made any discovery there. And so that's a reason why I connected it with V4641.
Not only was the microquasar V4641 Sagittarii the most likely producer of these gamma rays, the rays themselves had some incredibly high energy.
They almost the highest energy we could detect it from such object. It is about 200 TeV. It was surprised to find such high energy gamma rays.
A TeV is a unit of energy that stands for tera-electron volt, and the maximum energy that the team saw for their gamma rays was around 200 TeVs. But to make a gamma ray of that intensity, you need an even higher energy subatomic particle to create it. And it's not exactly clear what that particle is.
was the parent particle. Is that electron accelerated then produced gamma ray, or is that proton was accelerated?
To make a gamma ray in this way, you need fast-moving subatomic particles to smash into stuff. In this instance, V4641 Sagittarii has a black hole that is producing jets of matter that could be acting as natural particle accelerators, pushing a fraction of these particles to velocities that approach the speed of light, which strike other things and, bam, gamma rays. But as Xiaojie says, it's not clear whether the particles involved are accelerated electrons or protons, although she thinks the results provide more evidence for the latter. And if it does turn out to be accelerated protons, that could get physicists rather excited. Because of the way all this works, in order to create gamma rays at 200 tera-electron volts, the protons would need substantially higher energies in the peta-electron volt range. And something that could give particles this energy could be the long-fabled PeVatron.
So this is a particle accelerator that's capable of accelerating particles up to PeV energies.
This is Jamie Holder, who researches gamma-ray astronomy and who's written a News and Views article about the new work.
And there are some very good candidates, but there are no definitive sources that are definitely identified as a PeVatron.
And researchers really want to figure out what celestial object or objects could be PeVatrons, because they might help explain yet another mystery: the origin of galactic cosmic rays. See, I promised you we'd get back to them.
We're still trying to understand how cosmic rays are accelerated in our galaxy to the highest energies that they can reach. That's a 100-year-old mystery. The general way people would think about this is in terms of exploding stars. Supernova explosions have been good candidates for accelerating these cosmic rays. People haven't really considered microquasars, at least not recently. So this is kind of revitalizing this idea that maybe these type of systems could contribute as well.
And this potential microquasar source could be really valuable to scientists, because cosmic rays are really difficult to trace back to their source, as they can be pushed around by magnetic fields. But gamma rays aren't. So in this work, they might be acting as an arrow, pointing to where these mysterious galactic cosmic rays are produced. But of course, it remains unclear whether V4641 Sagittarii is a PeVatron after all. But Jamie was impressed with this new research and the technical achievement of identifying it as a gamma ray source, particularly given the location of the telescope that found it.
So Pork is in the north, in Mexico. And this is quite difficult for them to see because it's a southern hemisphere source. So the source never gets very high in the sky for them. And the plane of the galaxy, as seen from the south, has been pretty well mapped. So for them to see something new was a bit of a surprise. So one of the exciting things is that it tells us there's probably lots of other stuff out there to see.
For Jamie, there's a lot to learn about V4641 Sagittarii. And to do that, a lot more telescopes and types of telescope will need to be pointed at it to get a higher resolution understanding of the object, to figure out how and where gamma rays are made. Xiaojie says the same, and that in some cases this work has already begun. She also hopes that by learning more about how relatively near microquasars shoot matter out, researchers can get insights into their much more massive relatives, quasars, found in the center of distant galaxies, firing out matter across hundreds of thousands of light years.
So because they are too far away, we can only see a little bit dot on our map. So we do not have enough resolution to distinguish more detailed structures of that. But for microquasar, it provides us an opportunity.
Xiaojie Wang from the Michigan Technological University in the US there. You also heard from Jamie Holder from the University of Delaware, also in the US. To read Xiaojie's paper and Jamie's news and views, look out for links in the show notes.
Coming up, researchers have made an insulin molecule that's responsive to blood sugar. Right now, though, it's time for the research highlights with Dan Fox.
Comb jellies, found in oceans and seas around the world, may belong to one of the most ancient branches of the tree of life. But they have surprises left to spring. Now, individuals of one species have been observed fusing their bodies together. Last year, researchers spotted an unusually large, warty comb jelly with an extra mouth and sensory organ swimming around a laboratory tank. Suspecting this animal was a combination of two injured comb jellies, the scientists attempted to recreate this trick by taking portions cut from pairs of jellies and placing them next to each other on dissection dishes. In 9 out of 10 attempts, the portions fused together, quickly syncing up aspects of their nervous systems and digestive tracts. In the wild, this type of jelly rarely spends time with others of its species, something the researchers think might explain why these creatures seem to lack the capacity common in other animals to distinguish the tissues of others from their own. Read that research in full in Current Biology. A 2,800-year-old royal burial in Siberia includes what archaeologists think were people posed on horseback, after both the humans and the horses had been slain as sacrifices. The burial mound, called Tunug 1, is one of the earliest and most easterly sites with evidence of a culture similar to that of the Scythians, a nomadic people who later conquered much of what is now southwest Russia and Ukraine. In 430 BC, a Greek historian called Herodotus described how the Scythians used wooden stakes to pose dead men atop dead horses in an open-air tableau around the graves of their kings. Now, excavations at Tunug 1 suggest a similar display was created hundreds of years before this account. Researchers found horse and human bones, as well as pieces of horse-riding equipment decorated in the Scythian style. Positioning of the remains, along with pieces of birchwood that might be the remnants of stakes, suggest the sacrifices could have been posed as spectral riders, just as Herodotus described. You can find that paper in Antiquity.
Up next, researchers have created an insulin molecule that switches on or off depending on blood sugar levels. Synthetic insulin is life-saving for people with diabetes. This insulin, synthesized in the lab or modified from animal versions, enables people to control their blood sugar, stopping it getting too high, which can lead to coma and even death. But it also has a fatal flaw. There's a risk it can push blood sugar too low. That's because it's really hard to predict how much blood sugar levels are going to change, and synthetic insulin can stay active in the body for a long time in a way that natural insulin doesn't. So to avoid low blood sugar, many people with diabetes will be a bit conservative with their doses, meaning they may not be controlling their blood sugar in the best way, possibly leading to complications. Because of this, for decades researchers have been trying to develop an insulin-based molecule that can be more sensitive to blood sugar levels, switching on or off depending on when it's needed. It's been a tricky problem to overcome, but this week in Nature, a team from the pharmaceutical company Novo Nordisk present a molecule that appears to do just that. I reached out to the team behind the work, but unfortunately they were unable to speak to me. Luckily, I was able to get hold of David Sachs, a diabetes researcher who knows the paper very well, as he reviewed it and has been writing a News and Views article all about it. I called him up and asked him to tell me why it's been so hard to make an insulin molecule that can be sensitive to glucose.
Well, the main problem is that the insulin is basically switched on by high glucose and switched off by low glucose. And the glucose is maintained in a very narrow window. In healthy people and people with diabetes try to keep their glucose within a very narrow window, only between a little over 4 to 7 millimoles per liter. And that's very, very narrow. And so it's very difficult to have a molecule that can be switched by such relatively small changes in the glucose concentration.
But despite that, it seems that this week in Nature there is a new paper where they've made an insulin molecule that appears to be sensitive to glucose and is able to do this sort of switching. What was the approach they used in this paper to achieve that?
So yes, they took an insulin molecule and to one end they attached what they call a macrocycle, which is just a ring-shaped molecule. It's a fancy word for ring-shaped molecule. And this binds glucose. And they designed the ring-shaped molecule in such a way that it's specific for glucose and doesn't bind other sugars that can be found in the blood. And then, in addition, another part of the insulin molecule, they attached something called glucoside, which is a molecule that is derived chemically from glucose. And the way this molecule works is normally if there's low glucose, the glucoside, which is on part of the insulin, goes into this ring and the insulin molecule is folded over, so it's not active. And then when the glucose in the blood increases, it displaces the glucoside from this ring-shaped structure, goes into the ring, and then the conformation of the insulin changes so that insulin can bind to specific receptors inside cells. And these receptors can then signal, and the body knows that the insulin's there and the insulin's active and does its normal functions.
And what did they test this out in? Because I guess just showing it can do this in the lab isn't really enough.
They started off with actually doing experiments just in a test tube, used very sophisticated methods to characterize the molecule. And then they used cells, they grew cells in a petri dish and showed that it's active and can make insulin signal. And then they moved into rats and subsequently pigs. So they did experiments in animals, and the experiments were very promising and the results were very encouraging.
And what was your first impression when you first got this paper to look over?
Well, I think that this is conceptually a very big advance because people have been trying this for many years. Nobody's ever been able to achieve this success and show that it works so well, particularly in a pig model. Pigs are sort of a lot closer to humans than mice. In fact, the original insulin that was used in patients for many years came from pigs. One of the experiments they did is they actually injected it under the skin in the pigs, which is how people who have diabetes inject the insulin, the same place, under their skin, and they showed that it worked very well.
So this molecule then is sensitive to glucose, but as you mentioned, there's a very tight range of blood sugar levels that people want to maintain. Is this able to achieve that sort of very tight control?
Well, this is the first description of this insulin, and they did sort of fairly broad glucose concentrations when they were doing the evaluations. And so the range they used of glucose was 0 to 20. And as I mentioned, you're trying to get 4 to 7. So one of the limitations of the study is that they need to still document that it's sensitive over this very narrow range.
So I was wondering what you would like to see next from this field of research. What would be the best next step?
Well, the important thing is to actually do it in patients. It's going to take a while, obviously, the regulatory issues, but it's very important to determine in humans whether this is safe, which is the first thing. I mean, the results from the pigs suggest it could be, but it needs to be safe and it needs to be effective. And so the clinical studies need to be done to do this. If that's successful, then clearly it would have a big impact on patient care.
And I was wondering as well, a couple of weeks ago, there was a paper in Cell showing a complete reversal of diabetes with a stem cell treatment. And I know people are working on like a synthetic pancreas as well to produce insulin. And now there's this technology too. So altogether, I'm wondering, is this quite an exciting time to be in this field?
Very, very, very exciting, yes. I mean, advances are being made. I mean, you've also obviously heard, everybody knows about it, the new drugs that have very beneficial effects. People are losing weight because most people with type 2 diabetes, not all, but most of them are overweight. And when you're overweight, your insulin doesn't work properly. It's called insulin resistance. So weight loss can dramatically reduce insulin resistance. And people who have been trying for years to lose weight and have diabetes just have been unable to keep their weight down. And these new drugs, patients can lose 50 pounds, 60 pounds. And I mean, it's life-changing. So people have been waiting a long time for some of these advances. And I think the advances are fantastic in the last 5-10 years, really amazing.
That was David Sachs from the National Institutes of Health in the US. For more on that story, check out the show notes for some links.
Finally on the show, it's time for the briefing chat, where we discuss a couple of articles that have been featured in the Nature briefing. Nick, why don't I go first this week? And I've got a story that I read about in Nature, and it's based on a paper in Current Biology. And it's an ancient DNA story with a bit of a twist. It's about how hair found wedged in the teeth of a pair of famous lions that were killed in the 19th century gives us a sense of their diet.
Oh, wow. So hair can give us that much of a clue. But I was intrigued by one thing you said there, that these were a pair of famous lions. Can you tell me a bit about the background to this story? What lions were famous in the 19th century?
That's a very good question. And there are a few wild lions that are as well known as the man-eaters of Tsavo. And these were two large, maneless lions that terrorized workers constructing the Kenyan-Ugandan railway until they were shot by Lieutenant Colonel John Henry Patterson in 1898. Now, the exact number of people that these lions may have killed is unknown, but probably at least 31 near the Tsavo River in Kenya. And so, as I say, these lions were shot, and they ended up on display in Chicago's Field Museum. And this is where kind of the journey into the DNA begins, I suppose.
I mean, I guess they were infamous rather than famous, these lions. But my first thought, you said there was hair with some DNA stuck between these lions' teeth. Was this human hair?
Some of it was human hair, yes. So by 2001, thousands of hairs have been extracted from a cavity in one of the lion's teeth. There's a picture of it on the article over at nature.com. And at the time, I suppose, the best researchers could do was look at these hairs under the microscope. But as we've talked about on the podcast many times, ancient DNA techniques have come a long way. And where previously you'd imagine you'd need an intact hair follicle to take the DNA out of, what you can do now is actually get DNA from the hair shaft itself.
Oh.
Yeah, and so using these techniques, the team identified hair in the lion's mouth from giraffe, oryx, waterbuck, wildebeest, zebra, and of course human.
And so they're able to identify this range of different animals that the lion has eaten. What can this tell us more than the fact that they ate a lot of animals?
Yeah, I think it gives kind of a snapshot as to the predator-prey relationship. Now, the wildebeest apparently were a surprise finding, according to the researchers, because the closest herds are found currently about 90 kilometres away from where the railway workers' camp was. So either these lions roamed across larger areas, or historically, wildebeest did occur in this area. So it allows researchers to ask questions about animal behaviour and animal populations. But I think it's important to talk about the human aspect as well, because in the paper, the researchers didn't cover the information about the human DNA in much detail, and deliberately so, because of course there were people who were victims of these lions, and there are obvious sensitivities. And they say that any future work will need to be done with local communities and institutions. And one of the researchers behind this work is quoted as saying there are potentially descendants or a descendant community that may or may not want this type of analysis done, or maybe they do. We just don't know yet, end quote.
And so this is able to give us sort of a snapshot. What does this mean more broadly for our sort of understanding of, I guess, lions and this sort of analysis?
Well, I think ultimately the plan is to go even further than lions and use these methodologies, as I say, to look at ecology or the history of animals, perhaps even further back to extinct species. But one person that's quoted in this article says it's not particularly surprising, the list of sort of hairs that they found in this lion's mouth. But what it does show is the importance of preserving biological samples carefully, because they could be analysed at a later date by tools that may or may not exist at the current time, if you see what I mean. I mean, it has to be said, when this lion was shot back in 1898, it couldn't have even been imagined that this was something that could be done 100 and change years later. Who knows what we might be able to do even further down the line.
No, definitely. And my story this week is on that topic because it's a new kind of tool to look at DNA within cells. And interestingly, this allows you to look at DNA and also proteins without breaking the cell apart.
And for me, from being in the lab, I know that if you want to study DNA, you had to crack open the cell that it contained. In my case, it was bacteria, right? You could buy DNA extraction kits, they were called. But you're saying that you could sequence DNA without doing that?
Yes, that's right. So this is a combination of a couple of different known techniques that researchers have put together. So they give it the very snappy title, expansion in situ genome sequencing, and those words will all make sense in a second. So this was a story that was reported on in Nature, and it's based on a preprint. So caveat alarm, this has not been peer reviewed yet, so take it all with a little pinch of salt. But it could be a very exciting tool for researchers to use, and some of the people interviewed in this seem quite intrigued.
I mean, how does it work, Nick?
So the way it works, as I said, it's like a combination of a couple of known techniques. And one of them is there's a way to introduce an enzyme into a cell that copies DNA. And as it copies DNA, you can add fluorescently tagged DNA segments, so then you can see what it's adding as it goes along. And so using those fluorescent tags, you can be like, okay, oh, it's an A, T, C, etc. That is one part of it. But the trouble is, if you want to look at proteins as well, and you want to see the specific parts of DNA, all those fluorescent tags together, they're going to be really hard to distinguish as you're looking down the microscope, because the wavelengths of light will start to overlap. So what they've done here is they've done something called expansion microscopy as well. And this, as the name suggests, essentially expands the whole cell, so you don't get this overlap of the wavelengths of light because everything is further apart.
I mean, that's really clever. And so rather than taking the DNA out and doing this fluorescent sequencing in a box on a bench, it's introducing the machinery into the cell and getting it to do it itself. That is something.
It's really cool. And the really interesting thing about this is this expansion part as well, because I found it just weird to think about. But what it does is it introduces a gel that permeates the cells and then swells when it absorbs water, literally making the cell expand. And it does it in a similar way to how nappies or diapers, I guess, if you're American, expand. So there's a nice image for you. But yeah, as the gel expands, it just pushes the molecules further apart, and it makes it easy to distinguish one protein from another or one bit of DNA from another.
You did say that this is an unpeer-reviewed preprint, and this kind of seems too good to be true, right? Presumably there's more to it and the more work that needs to be done.
Well, there'll certainly be more work that needs to be done. And one of the things that is challenging about this is these techniques are very technically difficult to do. So many researchers interviewed for this seemed interested in doing it, but one of them is quoted as simply saying, it definitely looks complicated. So that may limit the number of people who can do it. But they have also tried this technique out to look at a particular thing. So they looked at something called Hutchinson-Gilford progeria syndrome. And this is a genetic condition that results in premature aging. So it's known that this condition is caused by mutations in proteins called lamins. And they were able to look at the DNA sequences and these lamins using this technique, and they were able to confirm previous results that had shown that these lamins, they seem to intrude into the nucleus and sort of alter the arrangement of the chromosomes, and that suppresses some amount of gene activity. And that's a similar sort of thing you see in people who are very old as well. So they're able to use this technique to look at interesting stuff, and this may be one area that is particularly useful for looking at the interactions between proteins and DNA.
Right, and in the complexity of a human cell as well.
Exactly, yeah.
Well, that is a neat one, Nick. And listeners, if you'd like to learn more about it and the lion story I talked about, look out for links in the show notes, where you'll also find a link to sign up for the Nature Briefing to get stories like them delivered directly to your inbox.
That's all for this week. As always, you can keep in touch with us on X. We're at Nature Podcast. Or you can send an email to podcast@nature.com. I'm Nick Petrichow.
And I'm Benjamin Thompson. See you next time.